Hi everyone. What's the term mutation means for you? What do you know about mutation? Now, let's explore mutation. For this topics you are going to explore, first the mutation classification and types, second, the gene mutation, and the last one is chromosomal mutation. 7.1 Mutation classification and types. There are 5 learning outcomes for this subtopics. First, define mutation. Second, classify mutation into gene mutation and chromosomal mutation. Third, define mutagen. Fourth, state the types of mutation. And state the types of mutagen. What is mutation? Mutation is a sudden random change in genetic material of a cell that potentially can cause differ in appearance or behavior from normal type. If mutation occur in reproductive cell, it may be passed from parent to offspring. Classification of mutation. Mutation can be classified into gene and chromosomal mutation. There are 4 types of gene mutation, which are base substitution, base inversion, base deletion, and base insertion. For chromosomal mutation, it can be divided into chromosomal aberration and alteration of chromosomal number. There are 2 types of mutation: induced and spontaneous mutation. Spontaneous mutation occurring in the absence of a known mutagens, usually due to errors in the normal functioning of cellular enzymes. While induced mutation is referred to a mutation which caused by exposure to mutagens. What is mutagen? A mutagen is anything that changes the genetic material of an organism. There are 2 types of mutagen. Physical mutagen such as UV rays, X-rays and gamma rays. While chemical mutagen includes colchicine and ethidium bromide. The sources of mutagen can be from sunlight, medical apparatus, food and preservatives, and also from cosmetics and cleaning product. 7.2 Gene mutation. Learning outcomes for this subtopics are: define gene mutation. State the 4 types of gene mutation. Explain 4 types of gene mutation. Explain base insertion and base deletion as a frame shift mutation. State the effect of base substitution resulted missense, nonsense and silent mutation. The effect of base insertion and base deletion to produce frame shift mutation. Explain base substitution which cause sickle cell anemia. Gene mutation is caused by a change in the nucleotide sequence of DNA within a gene. For example, as shown in the diagram, the nucleotide sequence in is changed. This resulted the change in nucleotide as shown in Q, changes in amino acid sequence of the polypeptide, thus different type of protein produced with abnormal function. There are 4 types of gene mutation: base substitution, base insertion, base deletion and base inversion. Base substitution involves the replacement of one nucleotide and its partner with another pair of nucleotides. In the diagram, it shows that in normal DNA sequence of the base of the 4th codon is cytosine. The cytosine is then substituted by thymine. This base substitution will change DNA sequences. Base inversion. Base inversion involves the change in position of 2 bases within the same DNA. Only one amino acid is changed from the normal polypeptide. The effect is not critical, such as base insertion and base deletion. Base deletion. Base deletion is loss of one or a few bases in DNA. One base in the normal DNA had been deleted. This will cause the changes in the base of the DNA starting from the deletion point. As a result, The amino acid sequence are also changed starting from the point of the base deletion. This will cause frame shift mutation. Base insertion. Base insertion is an addition of one or a few bases to triplet sequences in DNA. In the case shown here, the insertion of one cytosine base occurs in the original DNA producing mutant DNA. In mutant DNA, the sequence of base in DNA change from the point of insertion. Large scale of frame shift results in a new amino acid sequence, resulting the changes in protein structure. The effect of base substitution. Base substitution resulted in silent mutation, missense mutation, and nonsense mutation. Silent mutation. Silent mutation is an alteration in a DNA sequence that does not result in an amino acid change in a polypeptide. For example, guanine base in original DNA is substituted with adenine. This will change the base sequence in mutated DNA. But the amino acid produced is still the same. This shows that base substitution in this case produced the silent mutation. Missense mutation. Missense mutation is referring to the substitution of one nucleotide that change one codon to another, specifying a different amino acid. For example, the substitution of the adenine base instead of guanine, producing different codons specifying different amino acid. Thus incorporation of the wrong amino acid in polypeptide chain during translation. Nonsense mutation. Nonsense mutation is a mutation that change a codon into one of the stop codons such as UAA, UGA, and UAG. For example, the substitution of U instead of A in mRNA shown will produce stop codon UAG resulted, signaling the termination of translation. Frame shift mutation. Frame shift mutation is a type of gene mutation, where insertion or deletion of nucleotide causes a shift in the reading frame of the codons in the mRNA. Thus, may eventually lead to the alteration in the amino acid sequence in protein translation. Sickle cell anemia. Sickle cell anemia is an example of base substitution, which lead to missense mutation. Let's watch a video on sickle cell anemia.
What shape are your cells? Squishy cylinders? Jagged zigzags? You probably don't think much about the bodies of these building blocks, but at the microscopic level, small changes can have huge consequences. And while some adaptations change these shapes for the better, others can spark a cascade of debilitating complications. This is the story of sickle cell disease. Sickle cell disease affects the red blood cells, which transport oxygen from the lungs to all the tissues in the body. To perform this vital task, red blood cells are filled with hemoglobin proteins to carry oxygen molecules. These proteins float independently inside the red blood cell's pliable donut-like shape, keeping the cells flexible enough to accommodate even the tiniest of blood vessels. But in sickle cell disease, a single genetic mutation alters the structure of hemoglobin. After releasing oxygen to tissues, these mutated proteins lock together into rigid rows. Rods of hemoglobin cause the cell to deform into a long, pointed sickle. These red blood cells are harder and stickier and no longer flow smoothly through the blood vessels. Sickled cells snag and pile up, sometimes blocking the vessel completely. This keeps oxygen from reaching a variety of cells, causing the wide range of symptoms experienced by people with sickle cell disease. If the obstructed vessels supplied the brain, the patient could even suffer a stroke. Worse still, Sickled red blood cells also don't survive very long, just 10 or 20 days, versus a healthy cell's 4 months. This short lifespan means that patients live with a constantly depleted supply of red blood cells, a condition called sickle cell anemia.
In sickle cell anemia, the substitution of T instead of A in the DNA sequence will change the amino acid produced. Amino acid valine is produced instead of glutamic acid. As a result, the shape of red blood cell change from biconcave to sickle shape. Sickle cell anemia is one of a group of disorders known as sickle cell disease. In sickle cell anemia, the red blood are shaped like sickles or crescent moons. These rigid, sticky cells can get stuck in small blood vessels, which can slow or block blood flow and oxygen to parts of the body. Let's check your understanding for this subtopics by answering the quiz. Scan the QR code provided.